Students of standard 10th, we already completed cell division part 1, that is mitosis, which is called as equational cell division. Let's continue with this part 2, meiosis, called as reductional cell division. Now, let me tell you what are the contents in this. What is meiosis, its process, and how it is conducted in reproductive cells, carried on in reproductive cells. What is the significance of meiosis? How crossing over is defined, in the significance it is explained. Some important terms related to meiosis and difference between mitosis and meiosis. What is meiosis? Now, meiosis is called as reductional cell division. Let me tell you, at the end of mitosis we got 2 daughter cells from one parent cell, but at the end of meiosis we obtained 4 daughter cells. Now, this meiosis is called as reductional cell division, which takes place in all in the germinal epithelium of the gonads, which is composed of somatic cells, in order to form gametes. Now, during this type of division, the number of chromosomes in the somatic cells is reduced to half first, and then it is doubled, and then they are passed equally to the daughter cells, which are haploid in number. Now, the entire meiotic division is initiated and controlled by our endocrine activity, which is under the pituitary gland control. Now 2 phases of meiosis, as meiosis 1 and meiosis 2 we'll read. Now here it takes place it is mentioned already, it takes place in the reproductive organs, that is in the testis and ovary, to produce sperm and ova. It is also observed in flowering plants, where it takes place in the anther and stigma to produce pollen grains and ovules. Now here the cell division, the number of chromosomes in this cell division, the number of chromosomes halved and is termed as reductional division, because the number of chromosomes in the parent cell is divided into half the number in each daughter cell, hence the chromosomes are haploid in number. Now, meiosis is studied as meiosis 1, the first meiotic division, which is the reductional division. Let's see, it is continued as prophase 1, metaphase 1, anaphase 1 and telophase 1. Now let's see what is the prophase 1. Prophase 1 of meiosis 1 is very lengthy and complicated. It has many important events in this. There is a bouquet formation, pairing of homologous chromosomes, which we call it as synapsis. Then there is crossing over, then disjunction in metaphase. Now I'll tell you what is bouquet formation. The arrangement of all chromosomes of somatic cells in the form of bouquet at one corner of the nucleus. Now let's see what is synapsis. You can see here in the first prophase 1, there is pairing of homologous chromosomes, and there you can observe at the end there is crossing over. During crossing over, the genetic material present in the homologous pair is exchanged or transferred when small segments of the chromatids in a homologous pair are detached and attached to the opposite ones. Now, crossing over is a key event for variation among individuals born to the same parent, is a key event for variation. Now, next phase is which you observe is the metaphase. They are arranged on the equatorial plates, equatorial line, that is in the center. That is after disjunction of two sets of chromosomes are formed. Then this is followed by anaphase, which is moving towards the polar regions. that is, they move towards the opposite poles, during which spindle fibers shrink and they pull the half chromosomes towards the poles. Now you see the next is telophase 1. You see the chromosomes move completely towards the poles. That is, during late telophase they are surrounded by nuclear membrane. You can see again nucleolus starts reappearing. Then in a single, two nuclei are seen. It is followed by cytokinesis, where two daughter cells are formed with reduced number of chromosomes. That is followed by, again it is followed by interphase. Interphase may or may not be present. Immediately meiosis 2 begins. This is meiosis 2, the second part of the meiotic division, which is completely similar to mitotic division, which is called as equational cell division. Here at the end of meiosis we get 4 daughter cells. So let's see what is this meiosis 2. In this meiosis 2, again each daughter cell, each parent cell starts dividing into 2 daughter cells as prophase 2, metaphase 2, anaphase 2 and telophase 2, and which is same as meiosis, which is same as mitosis. So at the end of meiosis 4 daughter cells are formed, which are haploid in number. Now what is the next part is, what is the significance of meiosis? Now this helps in the formation of gametes. Here it is main significance is helps in the formation of gametes. Due to meiosis, the chromosomes of the somatic cells of any species is perfectly maintained here. The number of cells are perfectly maintained here, and it also provides a wide opportunity for variation. Here the chromosome number is halved, so that on fertilization the normal 2n number is restored. And it provides mixing of genes in 2 different ways, that is mixing of maternal and paternal chromosomes during the first meiotic division, during crossing over, as they separate from the homologous pairs. During crossing over they separate and they carry the genetic material. While they are separating, the chromatin material, which is very often gets exchanged between the 2 members of the homologous pair, resulting in genetic recombination once again. Now what is crossing over? It is the exchange of genetic material between homologous chromosomes that occurs during meiosis, and it contributes to genetic variability. And you can see the crossing over clearly. This is the first figure, shows the homologous pair of chromosomes. One is homologous pair, means one from maternal, this mother, and other on the paternal chromosome, which is having 2 chromatids each. There is each chromatid of a pair, they cross over in the second figure, and after detaching, that is non-recombinant chromatids are seen. That is, they detach from maternal chromatid, a part has been shifted to the paternal, and paternal chromatid, one part is shifted to the maternal chromatids, that is, we call it as non-sister chromatids. So during crossing over, the genetic material present in the homologous pair get exchanged when small segments of the chromatids in homologous pair of chromosomes are detached and gets attached to the opposite ones. Now crossing over is, as it is explained, is a key for variation among individuals born to the same parents. Let's see some important terms. These important terms are, what are homologous chromosomes? These homologous chromosomes are made up of similar length, structure, pair of chromosomes or having similar length, structure, centromere position, staining pattern, form genes with the same corresponding loci. Now what is synapsis? It is a pairing of homologous chromosomes that occur during meiosis. You can see in early prophase, you can see this, the pairing of homologous chromosomes. Mid prophase also you can see the pairing of homologous chromosomes that forms an X-shaped constriction over there, which is called as chiasmata, egg-shaped constriction between two non-sister chromatids. And again, late prophase, this egg-shaped constriction is still seen. Only in metaphase they move towards the metaphase, they get arranged on the equatorial plate, and anaphase they start moving towards the poles, they get detached there. Then chiasmata, it is an egg-shaped constriction formed due to crossing over between non-sister chromatids of a homologous pair of chromosomes. You can see the picture there, chiasmata egg-shaped structure over there between non-sister chromatids. Now let's focus on the difference between mitosis and meiosis. What is mitosis and meiosis? What is mitosis we have read, what is meiosis also we have studied. Let's see the difference. Mitosis occurs in somatic cells only and meiosis in reproductive cells. It provides growth and replacement of the dead tissues, and meiosis enables reproduction in both plants and animals for a specific period only. Mitosis, at the end of mitosis, 2 daughter cells are produced. At the end of meiosis, 4 daughter cells are produced. Mitosis, daughter cells formed are diploid in number, whereas in meiosis they are haploid in number. And during mitosis, daughter cells formed by single nuclear division, and meiosis, daughter cells are formed by 2 nuclear divisions. Here with we'll end up this chapter here with differences between mitosis and meiosis. Thanks for watching.